Now we are going to discuss about the evolution of these. Previously, we understood that Psilopsida had some outgrowths out of their aerial stem, which were photosynthetic but small and venless. Now, uh, when we come to see the radioactive or archaeological background of these coelopsids, for example, Cooksonia, we find out that these outgrowths were from where the leaves have been arising. So the arisal of leaves occurred 350 million years ago, 350 million years ago, and what actually drove that change was according to was actually unknown, and still there are two hypotheses which select which give the explanation of these things, which are the outgrowth hypothesis, and second is the reduction hypothesis. When it comes to the outgrowth hypothesis, it tells us that the first leaves which were arisen were due to you know um, a lateral outgrowth of the stem. These were the lateral outgrowth of the stem. These lateral outgrowth of the stem, since were on the lateral side, did not have the vascular bundles which were going upright to the stem. So, in order to get nutrition and water and minerals, they needed vascular bundles which were in the vertical stem, due to which in the second step, the vascular bundles start to develop in the death outgrowth. As a result of which, those vascular bundles being able to grow in that outgrowth caused to produce a definite structure which was green, visible, with vascular bundles named as single veined leaf or micropyle. This was the evolution of the first leaf, which we call as the single vein leaf micropyle, and that evolution started in the class Lycopsida. Example of this includes Lycopodium or Equisetum. It first occurred in which organism? Lycopodium, Lycopsida class, and after that, evolution of roots also occurred in line with evolution of leaf. Alongside leaf, there was evolution of root structuring as well. So the first leaves which were evolved were single vein leaves rather than the polyveined leaf of today's era. These were the single veined leaf which had only a single vein, no veins, no multiple veins were there, due to which these single vein leaves were known as microphiles. Micro, small or single, and pile for the word leaf, due to which these are known as microphiles. So this hypothesis, outgrowth hypothesis, is for the microphiles. But for there's another hypothesis for microphiles, evolution of microphiles, that um, these were formed due to reduction in the size of the stem. Now for that, we can just consider this diagram. Let's consider the diagram over here. Alright, so there isn't the diagram. So what we are going to do is, try to draw that structure on our screens. So um, it is told that previously plants had multiple branches, multiple branches of the same stem. Multiple branches of the stem. These multiple branches of the stem started to reduce. These multiple branches of the stem started to reduce, and as a result of reduction occurring, these branches being reduced were gradually reduced into a single veined leaf, which was known as microphyle, due to which a single veined leaf arrived. So this was a very very simple theory, a simple hypothesis, giving the explanation of how leaves evolved in microphiles. Now when it comes to polyveined leaves, these are the leaves which are present in seeded vascular plants as well as in Felicillophyta class, or in other words Pteridophyta. While when it comes to the single vein leaves, these are specifically found in Lycopsida, Sphenopsida, and uh, Lycopsida and Sphenopsida only. But when it comes to polyvein leaves, these are found in Pteropsida, Pteridopsida, class Felicillophyta, class Gymnospermy, class Angiospermy. Now, when it comes to when it comes to the evolution of polyvine leaves, these were evolved as a result. These were evolved from single vein leaves because it's an evolutionary line of origin. One gradually there's complexity going on in the structure. So in that sense, the next structure which would be derived would be polyvine leaves from the single vein leaves. Now for that, we have another theory which is known as webbing theory. Now let us understand that as well, that how this webbing theory occurs. Webbing theory basically has three major steps: overtopping, planation, and webbing or fusion. So when it comes to overtopping, overtopping is a stage in which it says that there is formation of multiple, in which there is formation of multiple multiple multiplanular multiplanular branches. There are multiple multiplanular branches, as you can see in the diagram. These are branches which are in different planes. These are unequal planes. We call those as unequal dichotomies, because we have previously understood that dichotomies are those branches which divide into two branches. So that structure is considered as to be dichotomously branched. Moreover, the structure of Psilocybin is also having dichotomous branching in its rhizome. Apart from, as it is shown in this diagram as well, that Psilocybin have dichotomous branching, particularly in the particularly in the group Rhynia, because group Rhynia belongs to Psilocybin, so it basically shows us that Psilocybin is having dichotomous branching. Now, as you can see within the dichotomous branching structure, it is an dichotomous branching of an unequal length, or we can also call it as unequal dichotomy. Unequal dichotomy. Now that unequal dichotomy or unequal length of dichotomous branches, when are formed on that leaf, this stage is known as overtopping, in which there is overtopping or unequal growth, growth of unequal dichotomies within the plant. After that, the second step which occurred is planation. Planation is a stage in which the unequal dichotomies are arranged on a single plane, as they were these unequal dichotomies which were present in different planes are now arranged equally in a single plane, which is known as planation. As you may see, this is a way the planation is occurring. Now, the third step which occurs is known as fusion or webbing. Fusion or webbing is a stage in which the spaces between the single planed branches are filled up by parenchymatous tissues, parenchymatous cells, causing to form a green blade around those branches, and that green blade around those branches is known as the lamina, which means formation of lamina starts as a result of filling up of parenchymatous cells into those blank spaces around the branches, as a result of which this causes to form us uh, causes to form us a multi-veined leaf. A lamina is formed. Now, as a lamina is formed, these branches basically work out as the veins of that polyveined leaf, due to which this structure formed over here results in the evolution of the first polyveined leaf, or the first megaphile. Polyveined leaf can also be called as megaphile, since what megaphile, these are certain terminologies which we need to remember. Megaphile, and that megaphile showed which type of venation. That was a reticulate venation. Reticulate venation because it has uh, network-like venation or arrangement of veins, due to which it was known as reticulate venation. Now, from the reticulate venation, there was evolution of parallel venation. And when it comes to the structure of evolution of leaf, the evolution of leaf took almost um, 15 to 20 million years. So it was not that simple. And when it comes to some examples in which this evolution occurred is Lycopodium and Equisetum. These were the plants in which the single veined leaves were present, and Equisetum as well. Now, when it comes to what we call as the polyvene leaf, we have example of this as Adiantum, Bryopteris, Pteris, Pteridium, as well as all the seeded vascular plants, which are examples of megaphiles. While when it comes to microphiles, we have only two examples as the living genera, named as Lycopodium and Equisetum. So this is all about the evolution. Hopefully, we'll meet again in another video.